Hello viewers, welcome to our channels discussing about cellular organization UGC CSIR net. So membrane structure and functions. What are the functions are present? Membrane structures, structure of model membrane, lipid bilayer and membrane protein diffusions, osmosis, ion channels, active transport, membrane pumped and mechanism of shorting. So membrane is a thin layer that forms the outer boundary of a living cells or an internal cell compartments. And molecular view of the cell membrane. Membrane consists largely of a lipid bilayer, which is a double layer of phospholipids, cholesterol and glycolipids molecules that contain chains of fatty acids and determine whether a membrane is formed into long flat sheet or round vesicles. Lipids give cell membranes a fluid characters with a consistency approximating that of a light oil. The fatty acid chains allow the many small fat-soluble molecules such as oxygens to permeate the membrane, but they repel large water-soluble molecules such as sugar and electrically charged ions such as calciums. So next slides on plasma membrane or plasma lamella is a biomembrane that occurs on the outside of the cytoplasm in both prokaryotic and eukaryotic cells. It separates the cellular protoplasm from its external environment. Prokaryotic cells do not have internal membrane portions and that occur in eukaryotic cells as covering of several cell organelles like nucleus, mitochondria, blasts, lysosome, Golgi bodies and perioxysomes, etc. These biomembranes, endoplasmic reticulum, they also occur on thylakoids inside the blasts or and outside the and inside the mitochondria. Vacuoles are separated from cytoplasm by a membrane called tonoplast. Chemical nature of membranes. Mostly the biomembrane consists of lipids 20 to 40% and protein 59 to 75% and carbohydrate 1 to 5%. Carbohydrate present in the membrane are branched or unbranched oligosaccharides. Example hexose, fucose, hexamine, etc. Proteins can be fibers or globular structure also. Next slides on lipid molecules are amphibiotic or amphipathic, that is, they possess both polar and hydrophobic natures. So, hydrophobic tails usually occur towards the center of the membrane. Protein molecules also possess both polar and non-polar side chains. Molecular structure of plasma membrane. All biological membranes including the plasma membrane and the internal membrane of eukaryotic cells have a common overall structures that are symbols. So the all cell membranes are dynamic fluid structures. Most of the lipid and protein molecules are able to move about rapidly. So next slides, the lipid bilayers. First indications that lipid molecules in biological membranes are organized in a bilayer came. The experiments were performed in 1925. This monolayer occupied a final area about the surface area of the original red blood cells because the only membrane in red blood cells is the plasma membrane. The experiments concluded that the lipid molecule in this membrane must be arranged as a continuous bilayers. The conclusion was rigid, but it is turned out to be based on two wrong assumptions that partially compensate for each other. And next slides, lamellar model, sandwich models. This is also they are early molecular models of biomembranes according to these models. For biomembranes are believed to have a stable layer structures. Antonio and Davidson model. The first lamellar model was proposed by James Daniel and Davidson in 1935 on the basis of their physiological studies. According to the Daniel and Davidson's biomembrane consists of 4 molecular layers, 2 of phospholipids and 2 of proteins. Phospholipids from a double layer. Phospholipids bilayer is covered on either side by a layer of hydrated globular or protein molecules. And next slides, this is also the lamellar models is picture also, and features of Davidson and Daniel model proposed a model called sandwich model for membrane structure, in which lipid bilayer was coated on it is either side was hydrated proteins. In this persons are discussed is about between molecules of different size and solubility properties, and also between ions of different charges. From the speed at which various molecules penetrate the membrane, they predicted the lipid bilayer to be about 6.0 nanometer in thickness. Thank you. Thank you. Please be subscribed our channels. Thank you.